So thanks for coming, everyone. I'm Cassandra Mitchell. I am the grant coordinator for InnovATEBIO, and today is the first part of our webinar series brought to you by InnovATEBIO, Bio-Rad, and the Center for Plant and Life Sciences at St. Louis Community College. We will be recording today's meeting, and the videos will be posted on InnovATEBIO website along with the slide presentations from our speakers. If you have not yet, please register for the second part of our series, and I'll drop that link for you right now. That's going to be on November 17th at the same time. And we are excited to now announce that the next part of this series will actually be an in-person workshop at the St. Louis Community College Center of Plant Life Sciences again. We're looking at dates in June and July, but we will be confirming that and sending out more information soon. So watch the InnovATEBIO newsletter, or of course, go to the InnovATEBIO website at our events page, where we post events like this and other things we have coming up in the future. And as this is an InnovATEBIO funded event, we of course will have a survey at the end, but the survey allows us to bring things like this to you in the future, and just helps keep the grant moving and the funds keep coming in. So please take that at the end. Also, at the end of that survey, Bio-Rad is offering free posters. So there's a little bit of incentive for you there. As we go through this webinar, please put all of your questions in the chat, and we'll have a Q&A session after each speaker. Myself and Dion will answer any questions we can about InnovATEBIO. Bryony and Abby will answer any questions about Bio-Rad, and I'm sure Betsy will jump in for any of the educational questions. And with that, I'll introduce Bryony, who has been instrumental in recruiting our speakers today and getting all these resources gathered up.
Thank you, Cassandra. So my name is Bryony Rug, and I'm director of the Bio-Rad Explorer program. And we've been working with InnovATEBIO and St. Louis Community Colleges for many, many years. And so I want to say thank you for helping us to put this on today. That's really great. And in particular, thank you to our speakers before you even speak for helping us to bring these technologies to our community. So I wanted to point out that while we can't cover everything about advanced PCR technologies in the next two presentations, I want to bring to your attention some other resources that are available to you if you want to know more. There are a variety of previous webinars covering many aspects of advanced PCR that we've done in collaboration with InnovATEBIO and St. Louis, as well as other folks. We have Bio-Rad bulletins on teaching real-time and droplet digital PCR, covering instruments and reagents. And the Bio-Rad Explorer program has 2 teaching kits to help simplify introducing real-time PCR to students. And lastly, Bio-Rad has a free full online courses for researchers on real-time and droplet digital PCR as part of the Bio-Rad Academy, which is on the website. And they go really deeply into the technology, how to set up assays and experiments and analyze data. And we will send links to all these resources to the registrants of the webinar. So you don't need to worry about that. They will come to you. So today we have 3 speakers, and we'll have a short Q&A after each speaker. We have Abi, who's going to give us a quick primer on PCR technologies, and Josh, who's going to tell us how he applies PCR in his coastal water research. And I will formally introduce those two before their presentations. And we have Betsy Boedeker, who's the district director for the Center of Plant and Life Sciences and the BioBench CRO coordinator for St. Louis Community College. And she will be the lead instructor at next year's hands-on workshop sponsored by the people here. Thank you so much, Betsy. So Betsy will lead the Q&A after the 2 speakers, and then she'll lead a discussion on strategies to introduce these applications to students. So please add questions or just discussion points into the chat as we progress through the morning, because that will help Betsy to work out what you want to find out at the end. So I would like to introduce Dr. Abiodun Bodunrin, and Dr. Bodunrin is an application scientist in the digital biology group at Bio-Rad, where he works to develop some assays for applications in oncology research, pathogen detection in wastewater, and infectious diseases. As an application scientist, he works with the marketing team to develop data and literature using droplet digital PCR. Abi is also involved in new product development, where he collaborates with marketing team, business development, and the research and development teams. He graduated with a PhD in biochemistry from the University of Houston, where he worked on characterizing universal stress proteins implicated in dormancy regulation in a gram-positive bacterium. And he got his Master of Sciences in Biotechnology from West Virginia State University. He enjoys long walks in nature in his free time. Thanks a lot, Abi. And if you could take it away.
Right. Good morning, good afternoon, everyone. If I could just share my screen. Okay. Can everyone see my screen?
It looks great.
Awesome. Okay. All right. As Bryony introduced, my name is Abi. I work as an application scientist at Bio-Rad in the presenting office. I joined Bio-Rad in the year 2020, December, after my PhD. And I'm here to talk about qPCR and ddPCR. Short introduction. All righty. Okay, so this is going to be an outline of everything I'm going to be talking about today. PCR overview. What is PCR? How does PCR work? I'm sure a lot of us are not strangers to PCR. So I'll quickly go over those, and then conventional PCR, talking about the instrumentation, analysis, the uses, and then I'll go on to qPCR, and that's the quantitative PCR, and then the ddPCR, which is the droplet digital PCR that Bio-Rad works on. All right, so PCR, this is just an introduction of the history of PCR. It's quite a phenomenal, innovation. It was invented in 1983. It was patented in 1985 by Karen Mullis, and it got the Nobel Prize for this in 1993. As part of the preparation for this seminar, I actually went back to read on the history of Karen Mullis, because it was discovered, he got the idea for PCR while he was on a camping trip with his girlfriend. And then his comments were that he has solved the most annoying problem in DNA chemistry in a single bolt, lightning bolt. And he said somehow he thought it had been an illusion. Otherwise, it will change DNA chemistry forever. These are his words. He said it's going to make him famous, and it was too easy. That was actually the part that struck me, that a lot of scientific innovations usually come as a result of serendipity, mistakes that are found in the labs. He said someone else could have done it, and it will surely have heard of it. That it's something that it's been being done all the time. So that was just something that struck out to me and came out to me. So in 1999, we had digital PCR that was described. It was actually found in 1990. And then in 2011, we had the droplet digital PCR branching out from the whole yardstick of the PCR. Sorry. Okay, so this is an overview of PCR. It's really simple. These are the basic components of the PCR. We have the DNA sample, which is usually the target. We have the primers, we have the dNTPs, we have the Taq polymerase that helps in the extension process, and we have the reaction buffer in which kind of everything goes in. It's a three-step process, which is just a recycling of this three-step process, which is the denaturing process, the annealing process, and then the extension. And this is done in multiple cycles, depending on whatever applications you want to do. So you could go from 10 cycles to 30 cycles to 40 cycles, but usually PCR is usually done in 30 to 33 cycles. And another application is the hot-start PCR. Now, the hot-start PCR is used in situations where you want to prevent primer dimers or non-specific binding. In this kind of scenario, you do not want a situation where you're preparing PCR in the lab or preparing your reaction or experiment and things begin to amplify. So they came up with this innovation of the hot-start PCR, whereby the DNA polymerase that is being used has been modified, chemically modified, to not make it work at room temperature. That's where the denaturing step, the first denaturing step, comes in, whereby you heat it at 95 for 10 minutes in order to break up all the bonds and can activate the Taq polymerase that is used, and which has been inhibited from working at room temperature. the initial denaturation step starts at 95 for 10 minutes, and that's where the hot start, and then it now goes on to the annealing process and the extension process for the PCR. All right, so this is looking at the DNA polymerase chain reaction. We have the DNA here, and then we have the denaturing step and the annealing of the primers to that particular step. So it's split up in the denaturation step. You have the two primers separated, which denatures them, and then you have the annealing of the primers to those two separated strands, and it is extended. For the first step, you have 2 copies. So it goes with this particular formula here, which is 2n copies. So for the first step, you have 2 copies. For the next step, you have 4 copies. The next step, you have 8 copies, 16 copies, 32 copies. In 30 cycles, you can have as much as 1 billion, copies of a DNA strand that you started with. Okay, so talking about the PCR, we're extending it to RNA because everything I've talked about is DNA. Now, how does it work for RNA? It's the process of the reverse transcription PCR. We use the power of the PCR to detect and quantitate RNA, and it must first be converted into cDNA. How does that work? We have a single strand with a poly A tail. This is usually found in humans that have a poly A tail at the end. You have primers that bind to them. So there are 3 steps. We have the oligo DT, we have the random primers, or we could have the gene-specific primers. It anneals to the poly A tail in this case. And then we have the reverse transcriptase, which is an enzyme that binds and converts that particular strand of either oligo DT, random hexamers, or gene-specific primers, and then it begins to synthesize. And then you can have your cDNA that can be used as a template in PCR. So instead of having a full strand of DNA, you could actually have RNAs that have been, extracted through mRNAs or microRNAs, and then that is actually amplified and converted into cDNA that can then be, amplified and, used for further downstream processing. All right, so PCR is the basis for ddPCR and quantitative real-time PCR. And this is just a table showing the types, the output, the applications, the benefits, and the pros and cons of each of this process. The conventional PCR, which is what almost everyone studied in college or high school, is a very simple process. It's endpoint PCR. So you start the process and then you use a gel to actually quantitate and look at the amplification that was done. This is usually based on sizes where you actually read it on a gel electrophoresis. And then we have the quantitative real-time PCR. It's been called by so many names. It's qPCR is also RT qPCR or RT PCR, RT being small letter. So it's just quantitative. In this process, you're not waiting until the endpoint. You're actually seeing the reaction in real time. What exactly is going on? How are genes being amplified? How are genes, coming up at particular cycles? So in that case, you can actually look at the genes that are abundant, that are coming up first in the cycle, in the amplification, and those that are not so abundant that are coming up last. I'm going to show an example of that, as we go further down the slide. The last one is the digital PCR, which is the droplet digital PCR in this case, or sometimes some other companies, Bio-Rad uses the droplet digital PCR, other companies use a micro or a nano well to actually partition this PCR reactions into thousands of reactions, and I will explain that in a bit. It's usually fluorescent detection, and its applications include genome editing detection, mutation detection of SNPs, copy number variations, and others like that. So we go on to the conventional PCR. It's been applied in, all right. So it's been applied in forensic science, in consumer genomics, food and agriculture, phylogenetics, environmental microbiology, and a host of other things like that. DNA fingerprinting, it's been used in the court of law, and other applications like that, thousands of them. And these are some of the instrumentations that we have for the conventional PCR. We have 4 different companies represented with Bio-Rad at the top level. We have Thermo Fisher PCR instrument, we have QIAGEN, we have Eppendorf. Here is looking at the top one is from Bio-Rad, and when I was in grad school, we were using the first one and the second one in my lab. But now that things have changed a little, we now have the PTC tempo that has just been launched by Bio-Rad. It's an advanced, form of that can do even much more, send your results to the cloud, and it's, pretty cool. Not like what I used when I was in grad school. All right. So here we have the quantitative PCR. It's real time. It's happening in real time. That's the name. And it's a technique whereby a target of molecule is amplified by repeat cycles of PCR. The same theory of the conventional PCR, except that this time around, you're actually looking at the results in real time. The amplification of the target molecule is monitored, and after each cycle of PCR in real time. So you're not waiting until the end. And in this case, you don't even have to wait. You don't even have to, do gel electrophoresis if you don't want to for this particular reaction. So here it's the curves that are coming up here are correlated with, so for instance, if the first, based on the abundance of the genes that you have your first curve that is showing up. So when you see the first curve, you know that, okay, that is correlated with an abundance of the genes that you have. The last curve being, oh, you don't have a lot of those genes expressed. So it's really, really very good when you're trying to do gene expression studies. In this, I did a lot of gene expression studies with qPCR when I was in grad school, and you're looking at the expression of this particular gene with this particular gene. How is it correlated with each other? The only drawback for the qPCR, which the ddPCR kind of helps to solve, is the fact that you don't have to do standard. You have to do a standard curve for the qPCR. In ddPCR, you do not need a standard curve. So this is an example of a multiplex PCR. So beyond single target PCR, you also have a multiplex where you're having more than one target that is being detected in a single reaction. The traditional PCR, you have one reaction in one tube, and that's what we see on the gel, looking at different sizes. For the multiplex PCR, it requires a careful design of your primers because you do not want targets that have the same sizes that can be seen on the gel. So you have amplicons of different sizes that are going to be detected, and that can also be done in a single tube with as many targets as you want. All right, so another example is the digital PCR, which I'm going to be talking about. This is just an introduction of how the Poisson distribution statistics is used to quantitate starting material in the digital PCR. What usually happens is that with a number of positive droplets that have been amplified, you correlate that with a Poisson algorithm that helps to give the concentration that you're going to be having in that reaction. But I'll talk more on that briefly. All right, so droplet digital PCR, what it is like. The way I try to explain this is that, for instance, in the lab, you want to carry out a reaction. You have 5 tubes, 10 tubes that you're trying to do PCR on. For the droplet digital PCR, what you're actually doing is you're separating all those reactions into 20,000 reactions that are going to be going on at the same time with the same volume of droplets. So you're having, instead of waiting 20,000 times to run 20,000 reactions, you're actually doing it at the same time in a single tube. That's the power of the ddPCR, of the droplet digital PCR. It has an increased sensitivity by increasing your local abundance. It also has increased precision, which I'm going to talk about the applications. So the key advantages of the ddPCR, like I mentioned, it has an unparalleled precision. The sample partitioning that you have that is afforded by ddPCR enables small fold differences in target DNA sequence between the samples. So in our case, you're having to do the PCR actually 20,000 times. So it's that kind of partitioning leads to high level of precision. It has an increased signal to noise, which is very sensitive. You can enrich for rare targets by reducing the competition that comes from high copy templates. So while everything is well separated, you can actually go in and pinpoint those particular rare targets that you're actually trying to study. As a matter of fact, they are highly complex ddPCR that have been found you can actually mask the effect of things that have a high copy number or high abundance, so that you can actually pinpoint the particular target that you want. And I would explain that when we get to the level of some applications of ddPCR. Okay, so this is how the workflow for Bio-Rad's ddPCR instrument looks. you prepare your reaction the same as you do for conventional PCR, but where it separates is in conventional PCR, where you take it directly to the thermocycler for your quantitative real time. You have a step where you generate your droplets, and generating droplets is you've prepared your reaction, and then this instrument helps to, we call it a droplet generator. It helps to generate 20,000 droplets all in a single well, and then you take it to the thermocycler. And then from the thermocycler, after you do your normal thermocycler, you now take it to read your droplets. So it separates out and reads your droplets. So you have positive, you have negative, you have double positives, and the likes. All right, so this is what the results for the PCR looks like after you've done your droplet reading. You have your positive droplets that contain. So here we have a 1D plot. And what happens is that each of those droplets that have been read, in some of those droplets, like I explained in the Poisson distribution, you have, in some of those droplets, there are times where you have one copy in them, or you have two copies in them, or you have no copy in them. For some of them that do not have any copy in them, that's what is separated into this gray cluster here. It's the negative cluster, which means it does not have anything in them. We have a thresholding that is done that separates the positive from the negative, that helps to actually takes this data, it counts the number of positives, takes it and correlates it with Poisson algorithm, and then you can actually get your copies, the concentration which comes out as copies per microliters. All right, so here is another example of the multiplex PCR. The one I showed was a 1D plot. This is a 2D plot showing in a situation where you've designed an experiment where you have two targets that you're trying to target. Two targets that you're trying to target. You have a FAM and a hex, which is the dye that is used. So the only difference is that you have a FAM that is, going after one target with its primers forward and reverse, and you have the hex, which is on the x-axis, that targets another target with its primers and that probe, and then you have the double positives, which is the duplex, and that makes it a duplex ddPCR. We also have higher levels of PCR instrumentation that Bio-Rad just launched, which is a 6-color, and I'll give an example of that in my application. All right, so like I was mentioning, this is an application where we have wastewater epidemiology, and I'll be leaving this to the next speaker to talk more about. We have wastewater application. So this was especially, relevant during COVID, where we were getting wastewater coming from stormwater intrusion, domestic wastewater, industrial wastewater from universities, from communities, and that can actually be analyzed for bacterial viruses. And then we designed assays that can actually detect this in 6 colors. So here we have targets in adenovirus, norovirus, and monkeypox, influenza A, that can actually be detected not just in 1 color or 2 color, but in 6 colors. So this is the power of the ddPCR that that other applications do not even have. The other application is found in liquid biopsies and mutation detection. Liquid biopsies are instances where you are actually analyzing blood samples and you're trying to detect cancer mutations in them. So in all blood samples, we have cell-free DNA, which is normal DNA that is shed into the bloodstream, and then we have the cell-free RNA. But found in blood samples of cancer patients, we have cell-free DNA together with the cell-free RNA. We also have the circulating tumor cells and circulating tumor DNA. So you can actually design assays that can go in and pinpoint if someone has a particular kind of cancer. It's some work that we're currently doing now in Bio-Rad, which is called MRD, which is the minimal residual diseases. We want to be able to detect cancer mutations in patients by sampling their blood samples, and before symptoms begin to show 2 years, or even after they've gone through a a post-operative procedure. You want to actually check if they still have those cancer mutations. So you want to check if it's been shed into the bloodstream. And then the other application is in rare event detection, where you're actually trying to detect, rare kinds of, targets or rare mutations in patients. This also applies to cancer detection, diabetes monitoring, organ transplantation, environmental studies, microbiome analysis, and the last application is in copy number variation. We can click on this link here to learn more about this process. And with that, I think that comes to the end of the presentation. If you have any questions.
I have one, which is you talked about the clinical applications, but I'm wondering how prevalent are those for patients yet? Because it's pretty new. So is this happening in the clinic yet?
So for ddPCR in the clinic, yes, it's happening. So in some instances, so for example, there's this, there are prevalent markers that are, being searched for. For example, in pancreatic cancer, in lung cancer, we have, EGFR mutation. The drawback is that you cannot do as many samples as possible, but in cases where you can do 2 targets, whether you're looking for EGFR T790M or EGFR L858R, which are, 2 of the most prevalent mutations in lung cancer, that is available, that is being used in the lab. And as a matter of fact, we've had cases where assays can be designed to pinpoint a specific or a particular kind of mutation, and that can be used to actually decide on the particular drug that is going to be, given to a particular patient, so to speak. But for MRD, we've gotten to that point where, and I don't want to, give out too much information based on some of the things that we're doing, but NGS is currently being used to actually sequence patient samples, and then we can then design assays that can be used based on those NGS data that we receive to, detect those particular kind of mutations.
And Abby, I might have just kind of bringing it back to some of educational perspective. A lot of our attendees are from high schools or community colleges. There's a few from university, but and so I know funding can be very limited in those environments. And so one of the questions that I kind of thought of is if they have primers that they're already using in some sort of conventional PCR assay, is that something that could be easily adapted to something in a qPCR or droplet digital assay using something like CyberGreen if they already have the primers?
Yes, that's actually very possible. Now that you mentioned funding, it just brings me back to my grad school days, the horror stories of not getting funded. I I only heard about ddPCR in theory when I was in grad school. I never actually saw an instrument because it's kind of a little bit expensive. But coming back to the issue of having primers already, yes, that can be adapted. You only need to do some validation, and we've been able to do validations for some of our customers that currently are using. They have primers, and they want to use CyberGreen, or they want to just get the Pro. We test that for them to actually find out if it works really well. So that's very possible.
And that's a lot of what we do with those 2 real-time PCR kits. We've done the validation for those kits. We substitute in a real-time, the CyberGreen reagent, so advanced reagent, and there's probably some small adjustments to protocol and things like that, but it pretty much work with primers that we have already. You do have to do the validation though. It's not just switch it out. You do have to do a little bit of work on the side, but not too much. Any questions from the audience before we move on? And we can always ask questions of Abi later on as well. All right. If we don't have questions at this time, then we will move on. I would like to introduce Dr. Josh Steele. Dr. Steele is a microbiologist at the Southern California Coastal Water Research Project, specializing in environmental microbiology, specifically identifying and tracking pathogens and naturally occurring microbes in wastewater, coastal ocean water, and coastal watersheds using cutting-edge molecular techniques such as next-generation sequencing and droplet digital PCR. His research focuses on developing methods to detect, track, and identify the sources of human-associated bacteria and viruses in stormwater and coastal water, developing and standardizing methods for wastewater-based epidemiology, applying molecular techniques to track antibiotic-resistant genes in wastewater and the environment, and using bioinformatics tools to link environmental DNA from natural populations to ecosystem health. He serves on the Wastewater-Based Epidemiology Committee of the California Water Quality Monitoring Council and received his PhD in biological oceanography from the University of Southern California, and joined the Southern California Coastal Water Research Project, which I think is called SQUIRP, in January 2014. Thanks a lot for presenting for us today, Josh. We really appreciate it.
Happy to be here. Thank you for inviting me. Let me see if I can share my slides. All right, can everybody see that?
Yeah.
Okay, perfect. So thank you so much. I'm going to talk to you all today about one of the ways that we use digital PCR and to look at beach monitoring and beach water quality monitoring in particular, and why we have been pushing these methods and going to these methods, and sort of give you a sense of an application in the environmental research world. There we go. So beach water quality monitoring programs, they've existed for quite a while. They exist in where we are in Southern California, and they exist in anywhere you have basically any sort of water that's used for recreational purposes as part of the Clean Water Act. And what they try to do, their main objectives are, they want to measure the beach water samples and then communicate the water quality. Now, I mentioned this has been going on for a while, and people have been doing this using culture techniques and, you know, with some innovations, but basically this is a 100-year-old technology of growing bacteria on a plate and seeing what you get, counting how many you get. So what's the problem with that? Well, the problem with it is that when you find that there are bacteria that shouldn't be there, they put up a sign like this. But if any of you have ever grown up bacteria, you know that it takes a while. So they go out and get the sample, and then they bring it back to the lab, and they try and culture it. And so that takes, you know, an overnight growth step. And so the next day, they put up this sign. But this sign is misleading. It's not saying that ocean water contact today may cause illness. What it's really saying is that the ocean water quality was bad yesterday. And if you swum, you might get sick. We haven't tested the water quality yet today, but tomorrow after we grow them up again, we'll let you know if it was safe when you swam today. So it's not ideal. It takes too long. In contrast, when we use a gene-based method like a qPCR or a digital PCR, we can get it much faster. So like I said, the traditional method where you count either colonies or you count wells after filtering your water sample and incubating them, it takes 1 or 2 days to get an answer. When we use qPCR or digital PCR, you can get that answer the same day, and you can actually post that beach and let people know that it's not safe to swim. When we have done it at the very, very fastest we could get it, we could get samples in the morning, and by that late morning or afternoon, we could let people know whether or not it was safe to swim there. So this is the motivation for these sort of newer techniques using a gene-based method, is it's just so much faster. So for those of you that aren't in the world of regulation and beach monitoring, or any of this takes a long time to get these methods changed. Like I said, they're using technology that's 100 years old, and people don't like to change easily. And so we started about 10 years ago, looking, actually before I started at SQUIRP, they were starting to look at qPCR for this. It's faster, just like Abi explained. You can measure the things, you can get them to light up and see what they are very quickly. They did a small scale study, and there actually had to be a law passed in California to allow them to start to change this regulation. We updated it and more recently did a study with San Diego County in Southern California, where they wanted to identify a new rapid indicator method, get the results on the same day. They wanted to rely on one water quality indicator, one bacterial target, and they wanted to be able to use this throughout their entire coastline that they had to measure, and then try it out and see how it went. we suggested using droplet digital PCR. This was it wasn't brand spanking new when we suggested this, but we thought this would be a good technique. And it has a few benefits over qPCR. They're already familiar with qPCR, but they're even more familiar with this culture technique. And so when we were explaining it to them, we liked this comparison. And this culture technique, some of you may have used it. We've certainly had high school and college students use this technique when we've worked with them, called IDEX, or Interalert is the name. And it's a culture method. You take 100 milliliters of water, you give it a medium that will light up if the particular organism is there and can metabolize it. And you pour it into this tray, which has a bunch of wells. And then you use most probable number statistics, count the number of wells that are positive, and you can calculate that using an MPN table to the number of the bacteria, the enterococci that are there. Digital PCR is similar. You have your sample in your PCR tube, you break it into 20,000 droplets, you count the number of positive droplets, you use Poisson statistics, not most probable number, but similar, and then you get your copies of target, and that can tell you how many bacteria are there. So that sort of, again, it tells you how digital PCR works, why we liked it, but there's more reasons why we liked it over qPCR too. Because you have that statistical technique of getting absolute quantification, you don't need a standard curve, which is what you typically do with qPCR. You have to have a reference for it to get a quantification. It also is more sensitive, has a lower limit of quantification. We can detect fewer gene copies per sample. And 20 gene copies in a 100 milliliter sample, that's pretty darn good. That's, just a few bacteria that we can detect. And even more importantly, what we found is that it's robust to inhibition. qPCR can work really well as long as there aren't inhibitory compounds in there that mess with that chemistry that's going on. Things that we often find in the environment, like large organic compounds or metals or anything like that, can really mess with your PCR, and digital PCR does a better job of handling that. And the final reason we chose digital PCR is we had looked at this before, and we'd done a comparison with culture and digital PCR in a slightly different water, in a stormwater discharge, and we found that they matched really, really well. So this was all really encouraging for us. So we went forward to try to do this. All right, for those of you who are into environmental policy, I'm going to quickly go over how we have to do this because of the requirements. That California law that was passed requires that we have an EPA-approved method or equivalent alternative method published by the EPA in order to replace the current method everyone's using. And the EPA requires that you compare this method at the sites you want to apply it to with a sufficient number of samples, which they don't tell you how many, but it has to be enough. And they have to cover the range of variation that you'll see, which they also don't tell you what that means. But they decide if you've got the high and the high concentrations and low concentrations enough to cover it. And then you have to have a strong correlation with the approved method. I'm not going to bore you with the details of what the correlations are. If you really want to know, I'm happy to tell you. But you basically, the simplest idea is just you need it to be a pretty strong correlation, an R squared of 0.6 or more. So with all that background out of the way, we have a method, and we wanted to do a study to see how it'd work and see if we could meet all these criteria. So we really took to heart how that we needed it at a sufficient number of locations and a sufficient number of samples. And we went, I think, as far as we could. We did 51 sample locations along the entire county of beaches. We had lots of different beaches, over 3,000 samples collected. We collected in wet or dry weather. And this was a nearly two-year study, and they sampled weekly, at least, sometimes more often. So this was a massive effort. And here on these maps, which are unfortunately a little bit blurry, sorry about that, but you can see we had a bunch of sites. This is actually going, A is the north coast of San Diego County, and B is the south coast. And so it goes quite a bit of coastline all the way down to the border with Mexico. So really quickly, again, we focused on Enterococcus. We wanted to compare two different methods, qPCR and digital PCR, and that culture and digital PCR. And it involved the County of San Diego, us, and the California State Department of Public Health, who were also interested in this. Okay, so with that out of the way, the first method we compared, like I said, was qPCR versus digital PCR. We got what we consider a great relationship, an R-squared of 0.76. Because we have so many samples, our p-value was amazingly low. And we had this shape, which is interesting. Just to make a note, we call it a broom shape to the plot. And what that means is, usually why that happens is that qPCR actually doesn't do as well at those low concentrations as digital PCR. And so you have more variability in qPCR when you start to get below a certain concentration. But at the high concentrations, they're right on top of each other. So this is something we've seen before. This was a fantastic result and really encouraged us to continue the comparison with the culture work. Now, one thing we had to fix with the culture work is that even though they're measuring the same bacteria, they're measuring something slightly different. When we're measuring genes, the bacteria may have multiple copies of genes. They may have varying copies of genes. And so we had to sort of scale the culture, which counts colonies, not cells, but colonies, to what we're measuring with the qPCR. And so we did that using what's called an intrinsic conversion equation, but basically it's what you see here. You have a curve and you try and sort of bring the scale on that y-axis together so that the curves can be more easily compared. And when we did that, you can see we had a very good correlation here. We didn't have a terrible correlation when we didn't, but we had a good correlation, so what we needed to do. So that was the meeting our goal here and what we tried to do in this study. Then the next thing they wanted was, they said, okay, great, we have a method, but how do we apply this method to the beach? What we typically do is we count a certain number of colonies, and we have a number, a threshold that if we're more, we have to post the beach, and if we're less, it's safe to swim. So we had to figure out how to do that. We went through a number of different tests and cut point analyses, and we attempted to maximize two things and minimize the false positives. So we wanted to maximize the percent agreement between the methods, and we wanted to minimize the percent of the false positives and the false negatives so that we could get something reasonable. And it ended up around with a threshold of about 1,413 copies. For what it's worth, the EPA's already approved qPCR method has a threshold of around 1,380 copies. So we were happy that we actually were coming out with a similar amount. So this made us feel really good. This made the EPA feel good. We got provisional acceptance. We published the paper. And just because we're very careful and nervous about stuff like this when we're trying to make new methods for people to use to tell whether or not it's safe to swim, because my kids go and swim at the beach and I want to make sure that it works, we compared to what we'd done before. And so what you can see here on this table is the study we did, which is digital PCR versus EnterAlert, and the range of R-squared we found versus previous studies. So study done in 2013 found a similar range. When we looked at digital PCR versus qPCR, we were right in the middle. Earlier studies found very similar ranges, whether we were measuring qPCR versus culture, whether we were measuring two different culture methods, we found very similar R-squared. So this made us feel good. This is about what we should expect. Environmental data is noisy, but we were getting a good range of correlations to make us feel confident. Okay, so we've got approval for use, and then they started using it. And once they started implementing it, suddenly everyone paid attention. They started posting more beaches. They started posting beaches at places they hadn't before because they had a more sensitive test. And that caused, as you might imagine, some consternation among people that lived or went to those beaches. And they also had some bad luck. They announced their implementation of this in May. And that summer, there was a lot of storms in the South Pacific, which pushes the ocean current up the coast. All right, so why is that a problem? Whoops, there. Well, we'd done a previous study where there is a sewage outfall down just a few miles across the border in Mexico. And it's a very basic treated outfall. It's just a settling pond, and then the sewage goes out. And it dumps, as you can see in this picture here, right across onto the beach. And when the current is going south to north, well, you can see from this model, this plume of untreated sewage basically goes up the coast along with it. Now, the culture method was catching it down here near the border, but it was missing it the further away you got. And so what you can see here on the right is a different target we were using, and this is what we call a human marker. So this is actually a bacterium that grows in about 75% of people's guts that we're able to measure. And you can see as we go from south to north, from left to right, from the sewage outfall up, we can see this pattern when we have south swells. We can measure it all the way up almost either to the most northernmost site we tested or almost. So again, this was bad luck. They announced it right when this was happening, and they had to shut a bunch of things down. You know, junior lifeguards and things like that got shut down, and people were upset. A lot of people wanted to know, well, okay, fine, you have this new method, but maybe the method's wrong. We had this old method that was working perfectly fine. How do they compare? So we had also done that in this particular case. We compared, so on the left you have the culture, and on the right you have the digital PCR, and you can see that for the most part, they have a very similar pattern. It's just that the digital PCR is more sensitive, and so the bars are higher. And so this looks really, so we have a really nice correlation again between culture and digital PCR for this indicator bacteria. And we were able to show them and provide the health department, who has to post the beaches, with some confidence that they were not messing everything up unnecessarily, but they were really showing something real and that it was protecting public health. And so I would like to say that the consternation has died down. That might be a little bit of overkill. It's just been muted a bit, but at least they have an explanation for what's going on. Okay, so all of what I've been talking about so far is we're basically measuring one, maybe two targets in a digital PCR. And Abi mentioned that the newer digital PCR and qPCR before it, you can measure 3, 4, and now with the new digital PCR machine up to 5 or 6 targets. This again is similar to what he showed you, where he was showing you the things that they measure in like wastewater epidemiology, which we've also been a part of. I think the last time I talked about COVID and that. But in this case, we, again, we're using different fluorescently labeled probes, and we can do multiple targets at once. And we're really excited about this, and I'll tell you why. As I mentioned before, we have this new method, and we like to use it for that indicator bacteria, and we have that human marker that we like to use. But we actually have a bunch of different things that we like to try and measure. So you see on the bottom here, we have our indicators, and Enterococcus is there. We also measure E. coli, especially in freshwater, for those of you who aren't on a marine beach. And coliphage is a new method. That's actually coliphage are viruses that infect bacteria. So they're the tiny little viruses in this picture all surrounding the big E. coli. And we like to measure pathogens so that we can know the things that are actually making people sick, because for the most part, these things on the bottom, while they're easy to measure and indicate some sort of problem, they're not what's making you sick most of the time. It's these guys, norovirus, you know, the SARS virus or any other viruses, or if you're unlucky to get these types of things, a Campylobacter or Salmonella infection, which are no fun. And that's typically what people get sick with when they're doing water. And then ultimately, as I mentioned before, we have that human marker. That's only one of the markers we have. We also look for other fecal contamination, dogs, birds, cows, basically anything in the watershed that could be contaminating that recreational water. We can look for. And then now that we are able to multiplex, we're working on trying to be able to look at many of these at once, as many as we can at one time, so we can more rapidly tell you what's going on and less expensively tell you what's going on. So we're really excited about this. And with that, I will leave it with this final thought, and you can see a lifeguard posting the beach. And where they ended up, I told you things were muted. They said that sewage blasted San Diego's beaches at a record pace in 2022. And what will the next summer bring? So I'm happy to take any questions.
So Josh, I have a question.
Yeah.
First of all, this is so gross. I'm glad I don't live on a beach. I'm just gonna, throw that out there.
Trust me, this is one of many examples of gross things on beaches.
I'm sure. So my question is, I definitely get that the droplet digital is going to be way more forgiving when it comes to inhibitors and the reactions. And that whole, that nano droplet sequestering of those different pieces of the reaction mix, I think is a big part of that. Mm-hmm. My question is, and so I know, like, for a lot of the stuff I do with droplet digital, do you guys have like a control that you expect a certain level of positives that you run for each of the samples that you're collecting, like kind of a known something that should be in everything kind of thing?
Absolutely. We do a couple of different positive controls. We can do, sometimes we do it with like a plasmid or a piece of DNA that we know the exact concentration of. And then other times we'll do things where, so for instance, in this case where we think it's sewage is the main contaminator, we'll use a sewage positive control too, so that we have an idea of how much all of these targets, all of these bacteria that we're measuring, or viruses or what have you, are in the sewage that should be going on right now. More questions? Bryony, I think
you're muted, Bryony.
I was saying people can type or they can come off mute. We don't have so many people here that it would become crazy. So if you want to just ask a question, feel free.
Well, I'm going to ask about negative controls then, since nobody else is chiming in. Do you have negative controls that you run as well to just kind of make sure that you're not seeing something that you shouldn't or should? Or
Absolutely, yeah. We have we will do, so we are very keen on controls, and we always run a template control. We also have and every time we extract samples, we have an extraction control that we do, a blank, as well as we sometimes do a spike in control. When we're feeling like we really need to understand what's going on with our processing, we'll spike things into the to a sample in the filtration step and in the extraction step and in the PCR step, different targets that we can do. So again, that's another use for the multiple, the multiplexing that we could do. So we can measure these all at once. And we also always run an extraction blank, a field blank, where they go out and make sure they're not just contaminating it by the people who are collecting the samples, and a blank when we process the samples, when we filter them. So yes, we have a lot of controls.
All right, Betsy, do you want to move into the next phase where we're talking about how you might use some of the information that we've been given from both Josh and Abby in for your students or in a teaching environment?
Yeah, sure. And I think one of the things that, again, I know a lot of our audience members are either high school level or community college for the most part. And I think and I think Josh, your presentation illustrated this beautifully, is that, one of the things that I definitely stress is the importance of some of those controls, because I think when you're doing a lot more like cookie cutter science where you're not, you expect it to work perfectly. Even a lot of the Bio-Rad kits that I use, they do have a lot of focus on the controls and the purpose of those, and what, the meaning of those controls is should you end up with a result that you don't expect. And so I think that's always one of the things that I really enjoy about teaching the qPCR class that I teach with our biotech program here, is that I think there's nothing that illustrates the utility of those positive and negative controls as well as qPCR or digital droplet for that matter, because I think it just really makes an impression. I guess, I love the examples, wastewater, there's just a lot of application to these different technologies. And I think that I love hearing about the different ways that they're being used. And even just like basically the whole validation that Josh and his team did to get this new droplet assay approved, and how you've got those regulatory agencies like the EPA who got to chime in on whether or not you can use it to make the determinations. I think that's really, really interesting. I guess I'm really curious how of the attendees right now, is there something in particular that you would like to talk about related to what you want to do with qPCR or droplet digital that may have not been addressed yet? And if there's any specific questions, or if you want to know the type of projects that I do with my classes, I'm happy to share some of that with you. I also love the inhibitors. I think is another one of those things that people don't necessarily think about off the top. And I think that is where I have a couple of experiments that we do where I introduce I actually just use some EDTA to inhibit the reactions, and the from like a dead flat line in qPCR, just illustrating how, your inclination is to add more sample when it doesn't work. But when you're talking about inhibitors, actually diluting the sample will give you a result where you can add as much as you want, but if the EDTA is still in there, it's not going to work. And by diluting it, you can, that makes a really big impression too. It's just by having a couple of, known samples, work well in an assay that works well, and just taking a little bit of that genomic and spiking in some EDTA, and then have them do it undiluted, and then have them do a 1 to 10 or a 1 to 100 dilution, and they see a result with those dilutions, but they don't with the straight sample. So, I mean, I think that is I have a lot of little things like that I've implemented in my classes just to illustrate some of those really key principles when you're talking about qPCR or droplet digital, just because it's really, it can be really, it's really, and this is what I say, and I'm sure Abby and Josh will totally agree with me on this. What I love about PCR is it is very simple. When it works, it's very simple, but when it doesn't work, it actually is pretty complicated. And there's so many things that you can mess with to try and fix it. So it's simple and complicated all at the same time. And I just kind of like that paradox of the two. And so So I'm from Shareth, you're interested in qPCR to identify antibiotic resistance genes from an eDNA.
I think Josh actually does this for his job.
Okay.
Right, Josh?
I do. I have done this. You will find you can and will find antibiotic resistance genes in eDNA. And for those of you that don't know what eDNA is, it's environmental DNA, although sometimes called extracellular DNA. I think that's what the original eDNA was, but now it's been broadened to anything from environmental DNA. And we do a lot with environmental DNA, and we do a lot with antibiotic resistance genes. Yeah, you can totally do this. I would recommend the more potential you have for sewage contamination, the better chance you'll have of finding them. But you'll find
so go to the beaches by you, Josh.
But you'll find them in soil anytime there's agricultural stuff too. You'll find antibiotic resistance genes. So I think that would be a great thing to do. We've worked with college professors and high school teachers who have done similar projects too. So you will totally find them.
What particular genes do you use for that kind of study?
Well, you can look, if you want to find the really, really widespread ones, I'd look at something at tetracycline resistance or something like that. But we've focused on the scarier ones for the most part, like the carbapenems, which are the antibiotics of last resort. Sort of, they're the ones they give you when nothing else works. And vancomycin, because they're very worried about vancomycin resistance. And we've done a lot of the MRSA, methicillin resistant Staph aureus testing. So those are the ones we've focused on. MRSA is going to be around a lot of places too. You could find that. College dorms, gyms, what have you. I bet you'll find MRSA there.
But for the widespread, which is probably somewhat better for a student project because you do want some success.
Yeah.
Maybe TET or MRSA would be.
Yeah, I think tetracycline resistance is one of the most widespread. So that would be one of my recommendations.
And I think also, some of my recommendations too, and I know Brenda, you have a question that I'm going to get to in just a second here, but there's oh my gosh, there's papers everywhere. There's proven work everywhere that I think if you just go in and do some NCBI searches, this not just with the environmental DNA, but I'm just in general, a lot of people have published data, and part of that publication involves primer sequences that they've used, and that just makes it a little bit less challenging if you can order primers that have already been proven to work. I think that's a great resource. I personally love to design primers, so I'll sit around and mess with stuff, and I'm usually doing gene expression work, so I have a little more stuff that I need to mess with for that. But I think looking at stuff that's already been published is a great way, using a proven primer set that you could implement into a class. I think is a great way to kind of have a head start on something working for your class. And so Brenda's asking about, Brenda, I'm not sure what CURE is.
Undergraduate research, course of undergraduate research experience.
Okay, gotcha. There's so many acronyms, I can't keep them all straight. And so I do know that here at my institution, a number of folks have successfully integrated something like that into their majors biology coursework. And so I do think it is actually quite feasible. I think, I'm happy to help you. I know Bio-Rad would be happy to help you. I'm sure there is some grants out there where you could maybe submit for some equipment.
I can add something to this, Betsy, and maybe Cassandra can, because InnovATEBIO has a whole section on cures Jim Hewlett is the co-PI for that part of the InnovATEBIO grant. So I don't know if Cassandra can speak to that, but I know that there's some stuff going on there.
Yes, we do have a lot going on there. And we have a hub. So I'll actually drop the link to that. And if you reach out to Jim, he will be able to answer more questions and help get started and that sort of thing. So he has more specific knowledge than me, but there's that link.
Yeah, so that's the student research part of the InnovATEBIO grant. And Jim has been doing that for a long time. Jim is at Finger Lakes Community College in Northern New York. And he also does stuff with freshwater sponges. So he would get very excited about that as well, Brandon. So I don't know what you're doing, if it's something like he was doing, but last time I visited him, he was extremely excited about freshwater sponges. So.
And then Craig is writing about his students performing cDNA inserts and then comparing that using restriction digest. We do something similar just with conventional PCR. We have a cloning unit that we do, and we try to use all of those things where we basically do a PCR, conventional PCR, that we then insert into one of the cloning vectors that are adapted to work with the PCR. You know how tactic polymerase likes to put that extra adenine at the end of a PCR product. And so you can there's lots of commercially available things that kind of target those PCR products to make cloning super easy, so much easier than 15 or 20 years ago, where you'd have multiple restriction digests and a whole cloning strategy. And it goes so much easier now. And so, yeah,
I can add some stuff in here as well, Betsy. So with the cloning, just if you wanted to do that, there is a Bio-Rad Cloning and Sequencing Explorer series, which is a whole course to make it easy. Our job really, the way we see our job, is to make this stuff easy, right? Yeah. So that's one thing. It's conventional PCR. There is multiplex PCR in there, but it's not qPCR or droplet digital. If you wanted to move into real-time PCR, we tried to give you some stepping stones. And so that's how we modified existing conventional PCR kits into real-time PCR kits. The most easy one that we have is based on our crime scene investigator kit. And really the crux of that is learning about doing a dilution series and then doing qPCR in your dilution series and being able to see that image that Abi put up of just the different cycles coming up with those different samples and being able to see, oh, I can actually see that there's totally different levels of DNA in this sample, because that's the power of qPCR or droplet, different from conventional. You can see how much is there. And so that's the basic one. And then there's the GMO kit where you can start to quantify how much GMO material is in your sample compared to how much plant material is in your sample, or not GMO plant or whatever, just regular plant versus GMO. So those are two real basic kits that allow you to start bringing these technologies in, because just to start from nothing is so hard, and we know that. And then you can go on and start reading a paper and pulling out primer sequences and ordering primers and getting all excited. But we know that first step is hard, right? So that's one of the reasons we made those kind of techniques and technologies. But Betsy, in July or June, whichever the date lands on, InnovATEBIO does the funding to bring people to that hands-on workshop. So definitely look for that, because we did one 2 years ago and it was awesome. People came in for 3 or 4 days or something. I don't remember how long it was, and just did PCR for 3 days.
It was super fun. We all had a great time. And so there's another question from Craig about the thermocycler. Is it special for RT, or is it the same kind for traditional PCR? So for droplet digital, and I know you guys the Bio-Rad folks can speak to this too. For the droplet digital, it is just a really good thermocycler, and then you're detecting the droplets after it's an endpoint detection. So you're detecting positive and negative droplets after the PCR is complete. When you're doing quantitative, you are actually detecting your product as it's accumulating in real time. So you do have special equipment. The qPCR is basically hooked to a good computer system that is reading your samples at every cycle and looking for that exponential increase in fluorescent product. You know, depending on what you're using, it could be cyber, it could be a probe-based system. But so you do need special equipment for qPCR, for RT-PCR, for even for the digital droplet, while the PCR itself might be in a regular kind of PCR, conventional PCR machine, you do have special equipment, the droplet maker, and then the detection that's happening after the fact would be required for that. But for quantitative PCR, yes, there is specialized equipment that is needed that is not the conventional PCR machine that you probably have, hopefully, in your lab right now. So, and I mean, if you guys, any of you that are going to attend the workshop over the summer, we have a variety of instruments here at our center. And I know last time Bio-Rad gave us a second one, just so for a demo so we would have more units to help process everybody's samples. So I think we have some great stuff that is on the horizon for that. Are there any resources?
Sanijba asked a question about designing experiments for qPCR. So if you want to go to that level, I would really recommend the Bio-Rad Academy. This is really in-depth courses. It's a course-based system. I think there's at least 5 or 6 hours of content for real-time PCR and equivalent for droplet digital, but it really goes deep into this stuff. Thank you, Abi. And it's made for researchers. It's not an educator resource. It's made for researchers, but it really does go deep into the kind of thing that you want to do. And it really does help. And you can also call Bio-Rad's tech support. Our tech support teams are pretty savvy in this stuff as well. I will say that, you know, if you have a question, they're very good at answering these things. So always use us as a resource as well. Betsy, do you have things too?
I am always happy to field questions or, help with primer design or troubleshooting. That's, again, one of my favorite things is when the qPCR doesn't work quite as it's expected, and then we get to troubleshoot it. And so there's lots of resources. I mean, the Bio-Rad little academy thing, let me just say, it's ridiculous. There is so much content here. I mean, and it's not just on, they've got stuff on flow cytometry. They have specifics on gene expression. And so, I mean, there is a lot of great content there that, I know all of you probably have lots of free time like I do. But I would love nothing more than to sit and go through some of their academy courses, just because there is so much content there. And I'm sure I always learn something when I sit through one of those. And so highly recommend all of that. And it's not just qPCR and digital droplet. There's a lot of other resources there as well. And so I love, Bio-Rad for that, because they just really do have just some great content that's going to apply for everybody. What was there something else I was supposed to say? Anyway,
just put the survey in there. So
yes, please take the survey, everyone. I'm happy to help with qPCR assay design if you want any help on that. I have a number of resources I could send. Somebody just reach out via email, and I'm happy to share any of my class resources with you guys. And I'm happy to share that if you come to the thing this summer. But I'm also happy to just share it with whoever. Yeah.
All right. Any last questions while we're here, or otherwise, I would like to say a really big thank you to both Abi and Josh and Betsy and also to Cassandra and Dion, who have helped us organize this event. And thank you all for coming and being interested and wanting to learn more about this advanced technology, which, you know, I didn't learn when I was in grad school because I'm pretty old. But I have learned since I was in Bio-Rad. But it's, you know, I think you should be proud of yourselves for trying to take it to the next level, because this stuff isn't easy and it takes a while to learn. So thank you all for coming. I hope to see you in 2 weeks' time, when we're going to talk about cell and gene therapy and AAV viruses in that. A totally different kind of talk, a totally different topic area. And we'll have a similar discussion at that point too. So hope to see you there. Please do the survey. It really helps InnovATEBIO maintain their grant funding, which allows us to bring this kind of thing to you. So please do that, and you can get your free poster as well.
Thanks, Bryony. Couldn't have said it better.